Hey there, biology. Let's talk about Unit 1 test. So, question 1, this is a picture of animal blood cells. A lot of problems we saw is that people were not reading the questions very well. So the smaller blood cells are red blood cells, blah, 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 blah. The larger cells are white blood cells, blah, 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 blah. The white blood cells have been stained with a dye. That binds to one organelle. This organelle is very important. Blah 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 blah. So the point of this, my reiterating that, is that the larger cells, these guys, are the white blood cells, and we need to measure the diameter of a white blood cell. So the idea was you pick the big ones because you're told that those are the white blood cells, and then you measure it. So it didn't really matter how precise you were on here, only to a certain degree. If you measured one of the red blood cells, you didn't read the question very well. But you should have gotten something between 1.2 to 1.5 centimeters. Whoop, there we go. And that was by design, because what you did is you said, okay, so that equals, okay, there's a cat, 12 millimeters to 15 millimeters. So that's your diameter. So they're between 12 and 15 millimeters, depending on which one you picked. The magnification power, remember, to find the magnification power, you'd use the measured diameter from the picture, diameter, divided by the given diameter, which is from microscopy. My cat is very interested in helping. And the point, what we were trying to get you to do is say, okay, so if the measured, which you got, was somewhere between 12 and 15, well, look, we told you the blood cells in real life are between 12 and 15 micrometers. So the idea was, if you decided, I measured a cell that was 13 millimeters, or 1.3 centimeters, I'm going to pick 13 to be the micrometers. You didn't have to, but it did make the math pretty easy. So let's say I did that. I measured a cell that was 13 millimeters, or 1.3 centimeters. Well, the first thing you have to do is realize that a micrometer, if you're going to do this division, if you're trying to find magnification, you don't want any units. The given unit is times bigger, right? So that's not a micron or a millimeter or a centimeter. So in order to get those units to cancel, you have to divide in the right units. So if I have a cell that's 13 millimeters, that's going to be, let's see, so 13 millimeters is, we know 1 millimeter is 1,000 micrometers, so that's 13,000 micrometers. Awesome. So then that's your measured diameter, 13,000 micrometers. So let's say I decided, then I'm going to pick 13 microns to be my measured one. Okay, so 13 microns, you just divide, you find it's 1,000x. Right? So that was the idea. If you picked, if you measured 12 and then you did it over 15, as long as you got something in the realm of 1,000x and you did your math right, you got credit. So let's say you measured a cell that was 15 millimeters, that's a millimeter, so 15,000 microns, and then you divided it by 12 microns, then you should have gotten a total diameter, or total magnification, of 1250. Okay, so that would be 1250 x. Okay, so the idea is, as long as you somehow converted something between 12 and 15 millimeters to micrometers, and then divided by something between 12 and 15 micrometers, you got credit for your total magnification power. Okay, so then the objective lens power, how do we figure that out? Okay, well, total magnification, while also being the measured diameter over the given diameter, equals your ocular times your objective lens. And we know that our total magnification was in the realm of 1,000x. We're told that the ocular lens is 20x, which is not what our ocular lens is on our microscope, so you had to actually read it. 20x times your objective. So then you would just divide both sides by 20x, and then you would get, boom, 100 divided by 2, 50, and that x stays in there just because, sure, and that is your objective magnification. Objective magnification. There you go. So that is the first page. So let's go on, and it's going to ask us about the things that were stained. So you didn't have to know what was stained, but it did tell you. The organelle in which the breakdown of materials, such as bacteria, viruses, and polymers, happens in animal cells. What is that? That is the lysosome. Done. The chemical process in which this organelle breaks stuff down is hydrolysis. Hydrolysis. So some people remembered that lyso means to break. Like if you use Lysol, you're breaking stuff down. Okay. That's also lyse here is in the word hydrolysis. Okay. So that's was kind of a little hint. And then what molecule in addition to the polymer is necessary? Well, there's your other hint. So hydrolysis involves water and your polymer. So if you have your polymer, let's do it this way. If we have some polymer, let's say it's a 2-monomer polymer, and you have water. Well, water goes in and it cleaves that bond to give you 2 monomers, and then water has been bonded to here. So that, you didn't have to say this stuff, you did have to say hydrolysis, and you had to say water to get the points for this. Okay, if bacteria and viruses are mostly made of proteins, what's the main type of monomer that will be produced? So proteins are made of, so protein is a polymer. The monomer of that, so what a protein is made of, are amino acids. Acids. Awesome. What chemical elements are found in this type of molecule? Sorry, that is carbon, hydrogen, oxygen, nitrogen, very, very important, and sometimes sulfur. If you put phosphorus, you lost points. There's phosphorus is not an important component in all proteins. It's there sometimes, but it's not always there. Carbon, hydrogen, oxygen, and nitrogen are always present in proteins. So that's what we were looking for there. We were looking for amino acids and CHON. Okay, so plants do not have lysosomes. In what organelle do they carry this out instead, or carry out this action? This is in the vacuole, which is, and animals do have vacuoles, but they don't do the same function. They do a little bit of the storage part, but they don't do the breakdown. So vacuoles, which also store nutrients and water and maintain the cell shape by being rigid, or being full of water, which keeps the cell turgid or full. Cell shape. If you put something else like cell wall, which protects the cell, or blah, 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 whatever, as long as you put, you lost a point because you didn't put vacuole, but as long as you put something that plant cells have that are not in animals, and then explained it properly, you got some credit. Okay, in question 2, we were looking for specific qualities of life, or properties of life, and an explanation. So the properties of life that we discussed were some sort of order, such as being made of cells, reproduction. We talked about how they use energy, they take in and use, anything that's living takes in and uses energy. There's growth and development. So what is that for? There is evolution or adaptation. There is, I'm sure someone's yelling it right now. What am I missing? Responding to the environment, to external stimuli. And then there's regulating the internal environment. Regulate internal environment. Now, some people put stuff like, your car exhibits a property of life in that it exchanges gases. Well, that's not a property of life. That's one of the many things that an organism does to regulate its internal environment, but that's not a specific thing. Some of the things we did see that were really good answers were, if you could say that cars have order because they have to be put together in a certain way, and it has to be like, in order for a car to work, it has to have some order to its engine, that kind of thing. That was reasonable. If you said, a lot of people said that they use energy, which is true. Cars take in energy in order to function. They take in energy in the terms of gas. There were some really interesting, so these are the nice obvious ones, right? So there were some, and regulating internal environment. Regulate, okay, can't spell. Internal environment. So that was one where a car can tell you, give you feedback if, or, you know, it takes in gas, does a certain thing. If you push on the brakes, it will lubricate the brakes, maintaining a certain kind of lubrication to keep your brakes working until it stops, etc. Any kind of internal regulation inside the engine would be a good example of this. Then we saw some interesting examples where perhaps cars evolve over time because it's driven by the market. So we pick cars that are safer, etc., etc. So that's kind of like a natural selection kind of process. We saw smart cars, people saying, well, what about smart cars that respond to an external stimulus, like beeping at you if you're about to hit something or stopping, that kind of thing. Nobody said anything about reproduction in cars, which if it had been justified, we would have taken, but is definitely not usually one people think of. So as long as you said something and gave a good example that clearly let us understand that you understood, let us see that you understood what these things meant, you got credit. For this one, you had to give 3 properties. This one, you only had to give 2. And so the most obvious one were the cars don't reproduce. And so cars don't make baby cars. That would be weird. Another one was, another one we usually saw, cars don't grow. That, you know, someone put a good example. That would be kind of awful to buy a new car and then in 4 years, you know, buy a sedan and then in 4 years you have a minivan. It's not really what you signed up for. Etc., etc. As long as, again, if you gave a good example, someone else put, let's see, cars don't grow, cars don't evolve, cars don't adapt. As long as you put one of those properties of life that we talked about and then explained your reasoning, you got credit. And you had to do this for 3 different things here. So we got a big range of answers. Some made no sense. And so if you think that you need to come and explain to us what you were thinking, you can try. Otherwise, though, as long as you put one of those 7 things and gave a good explanation, you got credit. All right, so for question 3, we have 3 cells here. They are not labeled. You didn't have to label everything. You had to follow the instructions. So on the lines below each, tell me if it's plant, animal, or bacterial. This one is plant. This one is animal. And this one is bacteria or prokaryote. So how do I know? This plant has a large vacuole. So you don't have to label this necessarily, but that's how I identified it. It has a cell wall that keeps it rigid, and it has a nucleus, which means it's not a bacteria. Nucleus. Over here, we know that this is also a nucleated cell, so this is and it's either plant or animal, and then I say, oh, there's only a cell membrane, and no cell wall, so it must be a plant, or sorry, an animal cell. It's not a plant cell. We also didn't really distinguish it yet, but these are chloroplasts. They have these flat membranes in them, and these are mitochondria. We see chloroplasts and mitochondria in plant cells. We only see mitochondria in animal cells. And then over here, which has basically nothing in it, that has to be a bacteria. It has a cell wall, so that's good. We know bacteria have cell walls, or they can. It has cell membrane. All the cells have cell membranes. It has no nucleus, which is one of the, why can't I spell? Nucleus, which is a defining characteristic of the bacteria. These are probably ribosomes. Most people label them that way. And then this is just DNA hanging out in the cytoplasm. So this is a bacterial cell. So that's what we were looking, really we were looking for this. And then your justification came in other questions. So which one is prokaryotic? Which one is eukaryotic? So the prokaryotes are the bacteria. Bum, bum, bum. You didn't have to justify. And then the eukaryotes, which means true body, which is talking in reference to the nucleus, are the plants and animal. Almost everyone got that right. There were a couple people who confused and put that the prokaryotes were the animals and the eukaryotes were the plants and bacteria. Plants and bacteria do share some characteristics, but the distinguishing characteristic between eukaryote and prokaryote is that nucleus. All right, one structure that's shared by all. Okay, so part C, which I totally read right the first time and did not do wrong at all, and then had to cut out of this thing. Part C, name one structure that's shared by all eukaryotic cells, but not the prokaryotic cell. So just one structure. There's many. There's the nucleus. There's the mitochondria. There is the, let's see, the nucleolus. There's the Golgi apparatus. There's the smooth ER. There's the rough ER. You could not, however, say cytoplasm, DNA, cell membrane or ribosomes, because those are shared by the prokaryotes, which is totally not what I just wrote by accident. So the nucleus, you had to state one of these things, of which there are many, and then give its function. Oh, you could have also said vesicle, vacuole, or lysosome. Okay, so let's just write that. Vesicle, lysosome, and vacuole. Hello, vacuole. All right. So you could have said any of those things, and you had to then give its function and label it. Okay, so if you said the nucleus, that is the control center for the cell, you had to label it in the plant and the animal to get full credit. Mitochondria is the powerhouse, it provides energy for the cell, you had to label that also. And so you could have identified the mitochondria because it's the bean-looking thing that's in both of them. Okay. The nucleolus is where ribosomes are made, and it's inside of the nucleus. You had to label both. The Golgi apparatus is what packages and sends off things like proteins and lipids. As long as you labeled it in both of them, you were fine. If you said it was this, yeah, it's probably the ER. This is more likely to be the Golgi. But we weren't so much worried of if you could identify this versus this. We were, could you identify something and say its function in both cells? Again, smooth ER, rough ER. Smooth ER creates lipids. The rough ER creates proteins that are leaving the cell. The vesicle packages things and moves them around. The lysosome is a digestive thing only in animal cells. Oh, so you couldn't actually have said this, or the vesicle, because those are just in animals. The vacuole is in both plants and animals, but you had to distinguish what it does in both. So in the vacuole, it's large and permanent in the plants and provides a lot of storage of water and nutrients. In animals, it's small and transient and can do some storage, but not long-term storage. So you had to, one of these had to be said and explained and labeled to get all 3 points. Okay. D, name 2 structures that are shared by all 3 cell types. So there are several. You could have said cytoplasm. You could have said cell membrane. Hello. Cell membrane. You could have said ribosomes. And you could have said DNA. DNA is more a molecule than a structure, but we'll give it to you. So, and you had to give their function, and you had to label it on the cells. So cytoplasm fills the cell, provides a liquid media for it, and the cell membrane separates the cell from the external environment and regulates some diffusion and osmosis properties. Ribosomes make proteins, and the DNA is the control center for the cell. It's the molecule of heredity in all organisms. So as long as you gave the function, you labeled it, and you named 2 different things, you got full credit for that one. So what's absent? Name 2 things that are absent from animal cells but are present in the plant cells. We have the chloroplasts, which convert sunlight energy into sugar. Well, they use sunlight energy to convert carbon dioxide into sugar. We haven't talked about it, as long as you said something like that. There's the cell wall, which gives cells their rigid shape in the plants and provide a measure of protection. There is the vacuole. If you put vacuole, you had to explain that it was the large permanent version of the vacuole, which acts as a water and nutrient storage substance. Is there anything else in the plants? I'm drawing a blank. Those are definitely the important ones that we've discussed. All right, so you had to put 2 of these and their function and label them on the plant, so distinguish them from the animal cell. Okay, so some cells have an extra structure or organelle that helps them move. What is this organ called, and what types of cells have it? So we were looking for the flagella. Okay, so the flagella. Okay, the most obvious example of the flagella is the sperm cell's tail. Okay, but so a lot of people just said this flagella, it's in sperm. Well, that's not necessarily true. It's in human sperm. It's in animal sperm, but plants have sperm too, and they don't have a flagellated cell. Okay, so you couldn't just put sperm. You had to say animal cells, and it turns out a lot of bacteria also have it. Okay, there's also a lot of protists that have it. We just aren't paying attention to that right now. But those are eukaryotic, and they're more animal than plant. We don't learn them. Don't worry about it. Okay, so if you said cilia, as long as you gave a reasonable explanation about how it's moving fluid around it, and mentioned again that it's in plants and bacteria, you may have gotten partial credit. Some people put something about vesicles or vacuoles. If you explain how it helps it move, because it might have been in the reading that it does, in fact, there is some kind of vesicle motion in certain cells. It's a very weird advanced topic kind of thing. You may have gotten some partial credit for that too, but the thing we were really looking for here was the flagella, which is in plants, or sorry, animals and bacteria, but can't be in plants because it can't go outside of the cell the cell wall. Okay, question 4. Question 4 is all about osmosis and diffusion, and you had to, again, read the question carefully. So the idea is we have some cell, and in each case, the concentration of sugar inside the cell, the concentration of glucose inside the cell is about 0.3 molar. Okay, so molar is a concentration. It doesn't matter if you remember what that is, as long you don't have to remember that it's moles per liter, as long as you remember it's a concentration. Okay, so in all 3 cases, we have a cell that's 0.3 molar. Okay, I drew that one kind of small. It doesn't matter. In all 3 cases, we put it in some sort of a fluid. So this is a beaker filled with some sort of a fluid. Okay, beaker, fluid. And then we want to see what happens to the water and the glucose. So in the first part of this, do we say that these cell membranes are permeable to water but not glucose? So in the first section of this question, the glucose cannot move. The glucose has to stay where it is because the semi-permeable membrane is only permeable to water, not glucose. So if this dermal cell is placed in a solution containing 0.1 molar solution. So remember we always talk about the concentration of something. We're talking about the solute, not the solvent. If you say 2% milk, you're saying there's 2% milk fat in it, not that there's 2% water. And we also never say it because that would be a solid almost. That would be just like a really hard cheese. And we never also say, we don't say 98% water because that sounds gross and weird and who cares, but if we say 2% milk, we know what that means. We're like, oh yes, that tells me what kind of milk this is. So when we say 0.1 per molar solution, we're saying there's 0.1 moles per liter of water, 0.1 moles of sugar, okay? So now we want to know what happens to the water. Well, everything in the universe wants to spread out as much as possible. That's the least energetic thing. Everything in the universe wants to be as stable as possible, and part of that means moving as far away from everything. So one way to think of this is if there's 0.1 molar sugar outside, there's 0.3 molar sugar inside. That means there's less water inside than there is outside. So our water wants to go inside. Okay, so water is going to move where there's more solute because that means there's less water. So the water moves into the cell. My dog is jealous. He doesn't get to do osmosis. He's whining in the background. Okay, the glucose cannot move. If the glucose could move, which is what's happening, stays. Okay, which is what the last question asked. How would each of the solutions be different if the cell membrane was permeable to glucose? Well, glucose wants to move where it's less concentrated, so the glucose would move out. Glucose moves out. Okay, if water is moving into the cell, and we don't care what's happening with glucose because it can't move, eventually so much water is going to enter the cell that it's going to balance somehow. So in either case, we're putting more water in the cell than was there before, so the cell expands. Or if you said explode, that's fine. The cell gets bigger somehow. Gets more volume. Okay, so the cell gets bigger. So in the next one, we have 0.9 molar. Okay, so if nothing else, you know that 9 is a bigger number than 3. So there's way more sugar outside than there is inside. Water's going to go to where there's more sugar because that means there's less water. So water's going to move out of the cell in this case. So water moves out. Moves out, so the cell is going to shrink. Kimball, shh, you're fine. Silly dog. So the cell shrinks or shrivels, either way you want to say it. Again, the glucose cannot move. Glucose stays. But if glucose could move, it's going to move to where it's less concentrated, so glucose would move in. Glucose moves in. Hello. Moves in. Again, everything wants to move to where it is less concentrated. Everything wants to spread out. If you walk into the cafeteria and there's a whole bunch of people at one table, you're not going to go join them when there's plenty of extra space if you are just trying to find some place to sit. Okay, everything wants to spread out as much as possible. Osmosis is weird because we don't look at things in terms of water. We don't say if this is 0.9 molar sugar in relation to sugar, what is it in relation to water? We don't care. We just fill it up with enough water to make it this concentration. So that's why when we're talking about osmosis or the specific diffusion of water, we have to look at the salt concentration, the sugar concentration, whatever it is, and do the opposite of what we expect, okay? So we know that water is going to move to where there's more concentration of the other thing. We know that glucose or anything else in the universe is going to move to where there's less. So glucose, if it is allowed to move, like in this last thing, is going to move to where there's less glucose. So in this one, glucose would move out. Okay, glucose moves out. In this one, glucose would move in to spread it out until it's even. Okay, you didn't have to say on the glucose part if the cell expands or not. Okay, what if you have an equal concentration inside and out? So if there's an equal concentration inside and out, a lot of people said nothing happens, the cell stays the same, etc. That's fine. Just know that technically you have an equilibrium. You have a balance of the same amount of water coming in as goes out. But the cell does not change shape. Cell stays same size. Hey, shush. Okay, and again, glucose cannot move. Glucose can't move. And really, water is moving in and out in equilibrium. Moving in, out, in equilibrium. But if you just said it stayed the same, that was fine. So, same thing here with the glucose. If glucose could move, which is in what's happening in this one, again, since it's the same concentration inside and out, the glucose would be in equilibrium, and it would look like overall it stays the same. So, moves in and out in equilibrium, or stays same. Okay, so it really looks like it's not doing anything when it's actually in equilibrium. Okay, that's all the questions. Please let us know if you have any specific questions that this little tutorial didn't cover. If you want to see pictures of my dog, just let me know. Bye!